Morning, guys. Hope you have your notes and a pen ready to go because today we're going to study the genome, okay? Today is all about DNA genetic technology, right? Remember that we've talked before about DNA being packaged inside of your cells in things called chromosomes. You have 23 sets of chromosomes, 23 pairs. That means you get 23 from your mom and 23 from your dad to make a complete you, okay? You guys have a full 46. Every single one of your cells has 46 chromosomes in the nucleus that has all of the DNA for your entire body. And the complete set of chromosomes in your body, that 46 chromosomes, is called your genome. All right? We looked before at these things called karyotypes. Remember, karyotype is just like a picture. It's a visual image of what those chromosomes look like from the first pair number 1, all the way down to pair number 23, and it gives you an idea of how to sequence them and organize them for some kind of genetic stuff. All right? And we've looked at these things before because karyotypes help you identify genetic anomalies and mutations. Okay? They can be stained, which is kind of cool. They can stain them, put some dye on them to let you know which ones have certain genes on certain parts, and it helps you organize how to collect them into pairs because some of them, it's obvious. This number 1, it looks pretty obvious that these two go together, but for number 1 next to number 3, it kind of could be a little bit confusing, right? One's a little bit shorter, but do they match? But if you take a look at how they've dyed them, you can see that there's a specific little stripe at the same spot on both of the number 1s and 2 matching stripes on both of the ones at number 7. Look at them and the correct sequence. Okay. They can also chromosome paint. They can paint or dye chromosomes. This is really, really important in the study, and they can paint certain parts. Like you look in the picture on the right, they can look for a very specific gene pattern, or they can just paint the whole gene. So you see that these 2 orange ones are probably number 2 or number 4, whichever chromosome they are. So they can paint them so they can organize them this way, or they can pull out specific little genes. Okay, that pulling out the specific genes is really important for helping them figure out genetic disorders. We've talked about genetic disorders before. We can talk about them some more, like hemophilia, which is a blood clotting disorder, or sickle cell disease, or Down syndrome, or Klinefelter syndrome. Those are all genetic disorders that they can identify through chromosome painting. Okay. There's a technique on chromosome painting called FISH, and this technique has a crazy name. I wrote it down for you. It's called fluorescence in situ hybridization. I'd rather just call it fish painting. And it has nothing to do with like fish in the ocean, okay? It just highlights similarities and differences between chromosomes, and it's really important in identifying new species of organisms, again with genetic disorders, and also it can tell scientists how closely related organisms are genetically. Okay, that's pretty cool. These guys, remember, are a few of the different ways that chromosomes can be mutated. Like, remember this one, when you take a part of a gene and turn it upside down, that's called an inversion. We're looking at the inverse. And over here, we have a translocation, where you take a section of one chromosome and try to transplant it onto another. These down here are another translocation. This particular thing is an insertion, where you take a gene out of one and then insert part of it into another. And then this one down here is a duplication. I think the one that I didn't see on here was a deletion. Okay, but remember, we've talked about some of these chromosomal mutations before, which can cause a lot of problems genetically for individuals. One way to study the genome, another way, is a gel electrophoresis. Now, gel electrophoresis uses something called restriction enzymes. Remember that an enzyme is something that makes a job go faster. It's a catalyst. It speeds up reaction. In our body, a lot of times, enzymes will break things apart, and that's called a catabolic reaction. It's breaking stuff apart. They can also glue stuff together. But in a gel electrophoresis, we're taking an enzyme and we're cutting a very specific portion of the DNA. And how it works is you create something called a gel, and this gel is has little bitty wells in it. They put a comb at the end of it, and then you can kind of with a micropipetter insert a little bit of DNA into each well and with the restriction enzyme in it, and then you see this kind of machine set up over here. It has a positive side and a negative side, and you run electrodes through it, right? And it makes the DNA kind of swim through the gel. And the basic theory of it is the shorter pieces of DNA will swim the farthest, and the longest will swim the least amount of distance, just because it's the ones that are short have less mass, they're lighter, and it's easier for them to swim, okay? And so based on the distance they're swimming, you can categorize the stripes that you'll see on the gel in like base pair distance lengths, okay? So you can see here there's a little image of a gel electrophoresis. Okay, on the next page, there'll be some more. This is a gel this guy is researching. You can kind of tell with these purple stripes here which pieces of DNA, if you look really closely, are more closely related than others. And gel electrophoresis is a really cool way to kind of identify, you know, relativity or relation in species. It's also a way to determine, you know, paternity. You can tell who someone's dad is based on looking at these things. Now, I know that we can talk about Maury Povich and I'm not the father or whatever, and that's kind of funny. But this is also a way for us to determine in forensics. They use it in forensics to determine who is the perpetrator of a crime, right? Or to make sure that the whatever blood or some kind of DNA they find at a crime scene belongs to the victim, which is kind of interesting, right? Okay. Like I said before, it'll help you figure out the paternity of someone or the perpetrator of a crime. Another thing we do with DNA technology is recombinant DNA. Now these little critters right here are called glowfish, and they are genetically modified. They are engineered to glow. So what has happened is some scientists have stuck some glowy or fluorescent bioluminescent DNA from a coral into these fish, and they start to glow, which is kind of neat. So recombinant DNA basically means you're combining the DNA from a couple of different species. All right, this involves some gene splicing, which we've talked before. So again, restriction enzymes can be used for that, gel electrophoresis. So it can also be used to take a piece of DNA out of one organism, like you see here at the top. We have this pink DNA, and we're cutting with restriction enzymes. We're cutting the little yellow snippet of DNA out, and we're removing it, and then we're implanting it or placing it into the green DNA right here. And remember that process of taking DNA out and gluing it back together is called gene splicing. So we've spliced this like glowy gene into the fish, and now the fish can glow. And the reason that that works is because, remember, like we said before, there are only 4 base pairs. We have adenine, thymine, cytosine, and guanine. And those 4 base pairs, those 4 nitrogen bases, are what every single living thing on Earth is made of. If you're a tree or an octopus or a rat or a bat or a cat or a dog or a frog, you are made of adenine, cytosine, guanine, and thymine, okay? And so that's the way that we can splice things into other organisms, is because the DNA is the same for every organism. All right. One of the ways that this technology can be used is that you can take technology that can or genetically modify an organism to be resistant to disease. You can implant some pesticide or insecticide that'll kill bugs on a plant, and then the bugs won't come and bother the plant, and then the farmers have a better harvest and we can have better crops, right? Another way that this recombinant DNA gene splicing is used is to help make insulin for people. We've talked before about diabetes being a disease that affected the way your pancreas or someone's pancreas made insulin, and insulin is important in storing sugars or keeping your blood sugar at a homeostasis. Remember that balance. So scientists have discovered that they can implant the human gene for creating insulin into plants or bacteria, and then they can harvest that when those plants or bacteria grow, and then that's one way that people can use insulin to help control their blood sugar. That's pretty fancy, right? The last thing we'll talk about today is something called gene knockouts. Okay, so here's how this works. You have tons and tons and tons of sections of genes and DNA and all those 46 chromosomes you have. And some of them are really, really obvious, their jobs, the jobs of certain genes or portions of chromosomes. And there's a lot of genes located on every single chromosome. And because the whole human genome, or most of it, has been sequenced out, most of it, we know that there are sections of genes or chromosomes we're uncertain. We're uncertain about their jobs or what they do or how they impact us. So how we determine the jobs of those genes, of those chromosomes, is we take a section out of an organism, like you'll see here, this mouse. So we'll take a part out and then we'll look and see how it grows, and we'll compare that knockout to a control group, another mouse that doesn't have any DNA removed, right? So you can see here in this image that we have this guy is a knockout mice, and we have knocked out one section of his DNA, one gene, because we didn't know what it did, and now we're trying to learn what that gene does, and we'll compare him to this mouse on the right who has all of his DNA, and you'll look and see what they discovered in this particular experiment was that that gene that they knocked out of this mouse on the left helped control weight, right? Helped control weight gain. So this way, these knockout mice have helped us learn a lot about DNA that we're not sure what it does. Okay, guys, thanks for listening in today and taking your notes over DNA technologies, and I'll see you on the next video or in class, and we'll talk more about genetic combinations. Okay, I'll see you soon. Bye.